First, I want to say thank you to all the organizers for putting together this wonderful event, and especially thank you to Jim for giving me the opportunity to give this talk today. As the title suggests, I'm going to be talking about how to improve biofuel feedstocks by modifying biosynthesis of the xylan. So first, how do we make biofuels? We take biomass, we pre-treat it, and then we use enzymes to degrade the cell wall material into sugars, and then we ferment them into usable biofuels. In our lab, we're mainly focused on how to make better plants so that we can make usable sugars in order to develop a more renewable feedstock for biofuel production. In plant biomass, the majority of it is secondary cell wall, which is made up of lignin, cellulose, and hemicellulose. Cellulose microfibrils form a matrix with hemicellulose and lignin, and this rigid cross-linking gives the plant its shape, structure, and protection from outside stresses and pathogens as well. Xylan is the major hemicellulose in plant biomass. It is made up of pentose polymers, and these pentoses are less desirable for fermentation because they don't ferment as efficiently as hexoses. So you would think that if we could easily just remove xylan from the plant, then this problem would be solved, but we can't easily do that because here you can see in xylan biosynthesis mutant, growth is greatly hindered when you compared to the wild-type Arabidopsis plant. In a normal wild-type vessel cell, they're round and they're large and round in shape. In the biosynthesis mutant, they're smaller and more irregularly shaped. And because xylan is important for water and nutrient transport within the plant, the collapsed vessels greatly inhibit the growth. Now, before I talk about how our strategy to xylan engineer these plants, let's look at the cell wall regulatory network in the wild type. We have specific transcription factors in the fibers and the vessels that regulate transcription of genes that make the secondary cell wall material. In the xylan biosynthesis mutant, there's a mutation in the xylan biosynthesis gene, and it no longer is controlled by these transcription factors. In order to reintroduce xylan biosynthesis into the vessels, we engineered a construct using a vessel-specific promoter, VIND6 and VIND7, to drive the expression of the defective IRX allele in the xylan biosynthesis mutant. Recall that in the xylan biosynthesis mutant, the growth is greatly inhibited, and you have irregular and smaller vessel cells. But in our engineered lines, we were able to recover growth fully compared to the wild type, and also rescue the vessel phenotype. But the main point is we want to see if there's still low xylose in these engineered plants. So we ran the samples on HPLC, and we see that our engineered lines still maintain the low xylose when you compare it to this wild type here. And we also looked at the hexose to pentose sugar ratio, and in all our engineered lines, we see an increase in this. Because the integrity of the cell wall was compromised in the background mutants, we want to be able to see if these plants will withstand normal environmental conditions if we were to apply this strategy to crop plants. And in order to do that, we measured the stem strength of these plants. And as you can see, a couple of the lines of our engineered lines were just as strong as the wild type. And we even have one line that was even stronger than the wild type. So we know that if they were to grow out in the fields, that they would withstand like very windy conditions. So with the increase in the hexose to pentose sugar ratio and also a decrease in xylose, we would expect that since there's less xylan in the plant, that it will be the cross-linked matrix would be less rigid and it'll be easier to release sugars from the plant cell wall. After hot water pretreatment and saccharification with CTEC2 enzyme, we see that all our engineered lines were able to release more sugars than the wild type. And this data is in per milligram of dry stem material, starting material. So in summary, in our xylan engineered lines, we were able to recover growth, and we were able to maintain the lower xylose amounts, and we saw an improvement in saccharification. But what now? How else can we make these plants better? I think yesterday Sam Hazen mentioned the importance of increasing the amount of biomass for biofuels. So that's what we're going to try to do next. We're going to try to increase the amount of biomass in these plants. And in order to do that, we're going to try to increase the fiber density inside these xylan engineered plants. So remember that NST1 is a fiber-specific transcription factor. So we made new constructs to drive the NST1 under the cellulose synthase promoter, and this creates a positive feedback, artificial positive feedback loop. And because the native function of the NST1 is to activate transcription of these secondary cell wall genes, the construct will have the construct will create an increase in the accumulation of NST1 transcription factor, which will in turn over-induce the expression of these secondary cell wall expression of these second cell wall genes. So we took one of our best xylan engineered lines and transformed them with the new construct, and I've begun screening them for thicker cell walls in the intervesicular fibers. So acting as our control is our original xylan engineered line. You can see that the fiber cells are pretty thin. I hope you guys can see that. But in our new densified xylan engineered plant, you can see much thicker cell walls in the fiber cells. I do apologize, I don't have a picture to show that the plant still grows just as well as the original background mutant and the wild type too. But we're also excited to see that we can stack these different approaches. So in summary, in our xylan engineered plants, we were able to recover the growth, maintain the low xylose amounts, and improve on the saccharification. And we were able to stack the approaches since we were able to apply the fiber density into the xylan engineered plants. And we still need to further characterize these transformants. And further down the road, we hope we can stack this with a lower lignin approach as well. I want to just acknowledge DOE for funding this research, and majority of the work on the xylan project I did with Pia Peterson, who also co-authored the paper with me. Fan, Pia, and Anandpat made the constructs. Jinsen helped me with the saccharification. Patanjali did the tensile strength test. Emily and Projecta helped me with the microscopy, and Barrett with the HPLC data. Sherry assisted me with growing the plants, and Henrik and Dominique provided support and advice through this project. Thank you.
Questions for Jane? You show increased sugar release
Yeah.
in your xylan deficient strains. I was wondering what percentage of that is actually due to increased, or I guess decreased cross-linking versus how much is linked to the increase in glucose to xylose ratio. So if there's just more glucose there or more cellulose, then you should be getting more release as well, independent of the cross-linking.
We actually haven't looked into that. So I'm not sure if it's due to the decrease in cross-linking or if there's just more, since there's a lower amount of xylose, there's an increase in other sugars to make up for that.